Hello, welcome to the Toxicity Assessment module of the Risk Assessment for Air Toxics course. I'm Ines Pagan, a toxicologist with the U.S. Environmental Protection Agency. The main aspects of toxicity assessment that are important for the overall risk assessment process are hazard identification or hazard ID in dose response assessment. The characterization of risk involves the derivation of a measure of exposure and a measure of toxicity. In this section, we're going to look at the measures of toxicity used to estimate risk. The process of characterizing the measures of toxicity used to estimate risk can be divided into two steps: the hazard ID and the dose response assessment. There are different types of data that are used in the process of hazard ID. Epidemiological studies are probably the most helpful because they tell us based on actual exposures to humans whether there is an increase in disease as a result of exposure to a chemical. We get this type of data usually from occupational studies and on occasions after accidental poisoning events. In vitro assays are usually performed in a lab using cell cultures. The person conducting a research will expose cells to a chemical of interest and see what happens. Very useful information can be gathered using in vitro tests. For example, the Ames test helps us evaluate whether a chemical can cause DNA mutations, and this is an indication of potential carcinogenic activity. In vivo assays are widely used to evaluate the potential toxic effects of chemicals. In these studies, animals are exposed to low, medium, and high doses of the chemical of interest. The results help us understand the kinds of health effects that may occur and at what levels. Researchers try to select animals with metabolic pathways similar to humans in hope that the exposed animals respond similar to humans when exposed to the chemical of interest. Experiments in humans can be conducted, however, it requires approval, supervision, and review by an institutional review board to ensure that these studies are conducted in a safe and ethical manner. Structure-activity relationship is the relationship of a chemical structure and the chemical's biological activity. This information can be particularly useful in making predictions on the toxicity of poorly characterized chemicals when we know the toxicity of a structurally related chemical. The first step of risk characterization is the hazard ID. Our main question in this step is there a causal link between chemical exposure and effect? Is the effect relevant to humans? And what is the nature and how confident we are on the evidence? So, we'll take a quick look at how animal studies are basically performed. In this example, we have 4 groups of animals, each fed chemical X in different amounts. The first group is fed a very high concentration. The second group is fed a medium concentration. The third group is fed a low concentration. And we have a fourth group that is fed no chemical at all, and this is what we call the control group. At the end of the study, animals are sacrificed to determine how this chemical has affected their bodies. Statistics are then applied to see if the health outcomes observed are statistically significant with relationship to the control group. In this example, the animals were fed the compound. In other cases, we have animals inhale the compound, and in other cases, we have chemicals that may be painted on their skin. The same chemical can cause very different effects depending on how it's administered, which shows that the route of exposure is a very important determinant factor on whether a toxic effect occurs and what the effect is. We also know that the amount of time over which a chemical is administered also plays a very important role in the type of effect that can occur. As an example of an outcome of the hazard ID is the EPA Weight of Evidence Scheme for Carcinogens. EPA has developed several schemes for classifying chemicals that cause cancer based on available scientific evidence. The weight of evidence narrative to characterize hazard of a chemical summarizes the result of the hazard assessment. This narrative also explains the kinds of evidence available and how they fit together. It also summarizes the mode of action, which is how the chemical acts in the body. The cancer guidelines have recommended descriptors for classifying human carcinogens. These are carcinogenic to humans, likely to be carcinogenic to humans, suggestive evidence of carcinogenicity, inadequate information to assess carcinogenicity, and not likely to be carcinogenic. So now that we know that a chemical is toxic, we need to understand how much dose of the chemical gives how much response. The hazard information is very useful and help us answer that question. So what dose response patterns are we likely to see? You can actually see a variety of dose response curves depending on the health endpoint you're looking at and the type of organism you're studying. For example, if you dose juvenile mice with a compound that affects the immune system, you might get a curve like the one you see here. This is called the sigmoid curve because it looks like the letter S. Adult mice, on the other hand, may respond with more linear curve in the same dose range. And in both of these examples, we're talking about dose response curves for mice being exposed to very high amounts of a chemical. As it turns out, our lab experiments on animals, also called animal models, only approximate what may be happening in humans. In the following slides, we're going to look at some of the methods the agency has developed to use animal data to estimate potential effects in humans. We'll start with non-cancer effects and then move to dose-response assessments for cancer-causing agents. In this example, a toxicologist is working on developing a toxicity value for chemical X. She goes to the literature to see what studies have been done on this chemical X and finds that 4 different studies are available. In one study, 3 different groups of mice have been exposed to different concentrations of chemical X, using a special apparatus that allows the mice to breathe through their nose and mouth. After exposing the mice for the bulk of their lives, the mice were sacrificed and autopsied. The results showed that different groups of mice had different amounts of fiber formation in the liver. Fiber formation is an indication that there is scar tissue forming in the liver, and this scarring interferes with the normal functioning of the liver. So, compound X caused liver toxicity. At the lowest dose, scarring was not observed, but scarring was observed at the high and medium dose. Liver toxicity was selected by the toxicologist as the critical effect, or the first significant effect observed as the dose of the chemical increased. It is important to note that in some studies, all doses tested show effects. In this case, the lowest observable adverse effect level may be used in the derivation of a safe reference level. As I mentioned before, animal studies can only approximate what is really happening in humans, and therefore there are many uncertainties that are addressed by the use of uncertainty factors when deriving the safe effect level or reference level. The derivation of the inhalation reference concentration involves a selection of a point of departure, which can be the no observable adverse effect level or NOAL, and if we don't have a NOAL, we would use the lowest observable adverse effect level or a low AL. And we would apply uncertainty factors to address differences between animals and humans, differences between humans, extrapolation from less than lifetime exposure, the fact that we don't have a NOAL and have to use a low AL, and an incomplete database. We will transition now to talk a little bit about dose response for carcinogens. We're coming to understand that some cancer-causing agents have a threshold like non-cancer agents. However, unless we have data to support this, we usually evaluate dose response of cancer-causing agents as though any dose, no matter how small, carries a risk that a cancer will develop at some point in the future. The higher the exposure and the longer the time, the higher is the risk of cancer. We also know from evidence in epidemiological studies that there is a lag time between the exposure and the occurrence of cancer. This time frame can be 10 to 40 years. For these reasons, we evaluate non-threshold carcinogens using a probabilistic approach. Here's an example of how we evaluate the dose response of non-threshold carcinogenic agents. Similar to the dose response curve for non-cancer, we have here 3 different concentrations of chemical X. Chemical X causes liver toxicity, which is a non-cancer effect, and brain cancer in mice. It is very important to note that these mice have been dosed with concentrations much higher than people would usually experience. Mice have to be dosed at these high concentrations because mice get tumors even without chemical X, and we have to give high doses to see any statistically significant change in mouse cancer rates. If we would have exposed mice to low levels, just like humans are exposed to environmental toxins, we would have to use hundreds of thousands of mice to see a statistically significant change. So, how do we extrapolate from what we know to what we need to know? And how do we do it in a way that is public health protective? One of the most common ways to make this extrapolation is by drawing the best line through the available data. Then we draw a lower level confidence level over the line, usually a 95% lower confidence level. At a point of departure at the lower end of the 95% confidence level, commonly a point where only 10% of the animals are responding, we draw a straight line to 0. The slope of this line becomes our dose response value for this chemical. For cancer-causing agents that are inhaled, the potency is given as a probability. The risk of getting cancer per unit concentration of the chemical in air in micrograms per cubic meter. This is called an inhalation unit risk, or IUR. For chemicals taken into the body by other routes, like ingestion, the value is given as the risk per milligram per kilogram per day, and this is called the oral slope factor. Carcinogens are considered non-threshold toxics. That is, there is no exposure level without cancer risk. Potency of carcinogens is determined by the slope factor. For inhaled carcinogens, we develop the inhalation unit risk, and for ingested carcinogens, we develop the oral potency factor. Non-carcinogens are generally considered threshold toxics. That is, there is an exposure level at which there is no adverse effect. Safe levels or reference values are developed for inhaled toxics, and these are called reference concentration. For oral toxics, they are referred to as reference dose. There are many sources for toxicity values, and you may find different values for the same chemical depending on where you look. In general, EPA gives highest priority to values found in its Integrated Risk Information System Database, or IRIS. This database includes information on chronic cancer and non-cancer toxicity values. Thank you for your time and attention. I hope you enjoyed this toxicity assessment module.